Hello everyone, and thank you for joining our webinar, Low-Pass Whole Genome Sequencing, Revolutionizing Genome-Wide Association Studies. My name is Brianna King, and I'll be moderating today's webinar. The webinar will be split into 3 sections, featuring Azenta Life Sciences Supervisor of Technical Applications, Dr. Elizabeth Louie, Gencove's Head of Data Science, Jeremy Lee, and Iris Kalka, who is a PhD student in Aaron Segal's lab at the Weizmann Institute of Science. A Q&A session with the presenters will follow, so please feel free to type your questions in the Q&A box, and we will try to answer as many as possible. Let's get started. In her role at Azenta Life Sciences, Dr. Louie provides consultation to our genomic solutions customers, including next-generation sequencing, gene and DNA synthesis, and any custom molecular biology needs. During her portion of the webinar, she will speak to the science and benefits of low-pass whole genome sequencing. With that, I'll hand everything over to Dr. Louie to begin her presentation.
Great. Thank you for the introduction. So, as mentioned, I'm going to give an introduction into low-pass whole genome sequencing. And for this section of the talk, it's called, low-pass whole genome sequencing, Revolutionizing Genome-Wide Association Studies. So first, I'll provide some background into different genotyping technologies that have been used over the last decade or so. Then I'll introduce low-pass whole genome sequencing and evaluating when to use it compared to traditional methods. And then I'll do a brief overview of some published applications over the last year. So in order to introduce genotyping technologies, I do want to stress the importance and why people are genotyping. So genotyping plays a major role in helping scientists correlate certain disease phenotypes with a genetic signature, which really allows to target certain treatments for development or gives a predictive implication in a genetic signature. And it can also be used in agriculture for breeding and strain selection. So many of the advancements that sample genotyping has undergone can be really attributed to a few key initiatives to capture the entire genome sequence. And one of them is, the Notable Human Genome Project. So this initiative spanned 13 years. It took about $2.7 billion and was a culmination of 20 groups worth of work. From this, 95% of the genome was sequenced. And the process itself was performed using a traditional Sanger sequencing route, which involved shearing of genomic DNA, generation of back libraries, identification of overlapping clones, the sequencing, and finally assembly. Documenting the sequence led to advancements in transcriptome profiling, targeted genome profiling, synthesis of novel DNA clones or products, and it opened up so many possibilities for targeted gene editing. It also led to other technologies that used hybridization approaches like DNA microarrays. So genotyping or DNA arrays are a cost-effective method to detect genetic variation in highly similar samples. It was a great alternative to what was previously a costly and laborious effort to sequence entire genomes all those years ago. And while limited to variants derived from existing data, there are many benefits that outweigh that limitation, such as the cost, the speed, and its accuracy. Microarrays allows for studies of thousands of patients, it's high throughput, and it can efficiently detect known SNPs at high accuracy. However, there are some limitations to all of that. So the method is heavily reliant on known SNP data, and arrays and panel probe designs have been biased to European populations. Despite this, genome-wide associated studies have continued to grow, though the focus, as shown in this graph here, is still heavily targeted to that one population. Therefore, while there are time, cost, and accuracy benefits to using the arrays, the applications of the studies are limited to better studied populations or to the detection of those known variants. These limitations can really only be overcome through DNA sequencing. Now, the cost of sequencing has continued to decline since the Human Genome Project completed, with significant strides with the introduction of high-throughput screening options like NGS, and it dropped significantly with the introduction of the first Illumina NGS platform. The cost has continually gone down due to additional platforms being developed and new technological advancements, and the number of sequencing publications has accordingly increased. The graph here highlights 3 key initiatives: the 1,000 Genomes Project, which documented hundreds of human genomes; the Cancer Genome Atlas, which documented several thousands of tumor normal genome pairs; and the Exome Aggregation Consortium, which documented over 60,000 human exomes. Many of the data sequenced to date have been whole exome rather than whole genome, but as advancements are continually made, we do expect that the ratio to be increasingly favored towards whole genome in the future. Now, nevertheless, while whole genome sequencing overcomes that bias of arrays, and while the costs continually decrease and drives adoption, it currently remains price prohibitive for larger population studies for researchers and groups. It's important to understand the pros and cons of certain DNA sequencing workflows so that scientists can weigh the benefits towards their specific project goal. So I'd like to review the advantages and disadvantages of the whole genome approach, as well as comparing it to a targeted sequencing approach like whole exome sequencing. So whole genome sequencing is still likely one of the more versatile genotyping options. It allows detection and analysis of samples without a known or complete reference sequence. It also provides detection of different abnormalities beyond the typical variant detection, including structural rearrangements and copy number analysis across the full genome. Appropriate applications would be anything that requires that discovery element, or where a reference sequence isn't available or incomplete, such as a biomarker discovery experiment, if you're doing contamination testing of products, or any type of novel organism study. Small genomes could also benefit from using whole genome since it doesn't really require a lot of data. On the other side, there are targeted sequencing methods that are frequently used. So this can be targeted through a PCR assay or through a probe hybridization method that enriches for either a SNP or a region of interest. With either approach, though, unlike whole genome sequencing, the design would require known genomic sequences. I would recommend one approach over the other when it comes to targeted. And that's based on, number one, the length of the region or regions of interest. So using a probe approach to capture a full 40 kb gene is likely going to be easier than designing multiple overlapping PCRs to cover that same region. A second point of consideration is the availability of unique sites for primer binding, such as secondary structures or high GC-rich segments that surround that region of interest. That might not be conducive to a PCR amplification design. And lastly, whether or not one of the methods would introduce an unintended enrichment bias due to that method that's being used that prevents you from reaching your project goal. For example, if you're interested in novel variant identification or you're interested in truncated variants, you might not want to use any preconceived data that you have that will run the risk of you missing any new targets that you're actually looking for. Now, targeted sequencing has many benefits. It does provide the ability to sequence low frequency variants with a deep coverage and with a lower output compared to performing like a whole genome approach. It's quite robust, so it has a focused and easily interpretable variations for disease models to inheritable disease. And if you think about the human genome, 2 to 3% of it actually encodes for the protein, and many disease biomarkers are located in that coding region. So therefore, targeting the select regions or even just the exome allows a researcher to hone in on relevant regions for sample comparisons without getting distracted by all of the other data that's there. But that said, the exome and all the coding regions is really only just that 2 to 3%. the remaining 97 to 98% would not be captured. And therefore, you're going to be losing a lot of information. So since many pre-developed panels and assays are heavily geared towards those coding regions, you're going to miss things like certain abnormalities, like large substitutions or deletions in the genome, potentially large insertions, and you'll also miss these chromosome translocations. So therefore, to summarize the limitations of both approaches, whole genome sequencing overcomes the bias of a targeted approach, but would require a lot of data to get that same amount of coverage and detection sensitivity. The deeper sequencing requirement therefore limits the feasibility of having a high-resolution, large cohort study. The targeted approach, on the other hand, is limited to that understanding of the genome and exome, and therefore any mutations that kind of fall outside of that could be missed. So this really brings us to today's topic, low-pass whole genome sequencing. So low-pass whole genome sequencing is a high-throughput solution that's comparable to that traditional genotyping method like a microarray. So sequencing is performed at low coverage, anywhere between 0.4x to 4x, and uses imputation to generate the variant call. As a general overview of imputation, it uses statistics to make calls and fills in any missing data with a predicted sequence based on a database of genome information. Imputation allows us to actually provide up to 99% accurate variant calls across that genome. So let's take a look at how imputation can compare to the other technologies. So if looking at a array comparison, in a study released last year, data from two populations consisting of individuals from a historically more researched population of European ancestry was compared to individuals of African ancestry, which is historically less common in association studies. For experiments A through D that's listed here, individual samples were sequenced anywhere between 0.5x to roughly 1x coverage. And for the last experiment E, the same was tested on a global microarray. The results on the left show the non-reference concordance across the different experimental groups. Genotyping concordance is a metric for imputation accuracy. So it shows here that low-pass sequencing plus imputation provides accurate calls at effective coverages of 0.5x and higher, and the results were comparable or better than the array data. Particularly in the African ancestry group, that did benefit the most out of this type of study. So making low-pass whole genome sequencing as part of the study does make it a viable alternative to a traditional array. So whole genome, low-pass whole genome sequencing can be compared to the different platforms in a few different ways. If we look at cost and coverage, there is quite a few advantages. So the relative cost is comparable to an array and is lower than whole genome and whole exome, and can be lower than a potentially targeted solution as well. So when it comes to novel SNP detection, low-pass is a practical method compared to whole genome and whole exome. And for the human genome, imputation does allow for interrogation of up to 85 million SNPs for variant detection. Structural variant detection, on the other hand, does rely on good coverage across the entire genome. So low-pass coverage is not ideal for that type of detailed analysis. Though in the recent months, some studies have used low-pass whole genome sequencing as a screen to filter out specific samples for that deeper testing. And copy number variant detection is still possible with the shallow read depth that low-pass provides. So thinking about some of the benefits that are highlighted here, low-pass whole genome sequencing does allow for high throughput processing due to the low data requirements, and is an unbiased detection method when it comes to working with less studied populations, and it does allow for off-target analysis. Therefore, it's well suited for genome-wide association studies, and especially advantageous over arrays when it comes to those underrepresented populations. Low-pass whole genome sequencing can be used for phylogenomics, for breeding studies, cell bank profiling, and even patient testing for any type of risk outcome assessment. Low pass should be avoided for use in any genomes that are not well characterized or for any type of de novo assembly. Now, some groups and some researchers have used low pass whole genome sequencing to build their database, but if you really need a ready to interpret solution, it's not going to be the ideal method. And while low pass can be used to detect SNPs, the low coverage should not be used for any low variant frequency detection studies, and it should also be avoided for making calls on patient disease prognosis or diagnosis. So keep these questions in mind when you plan your experiment. Is there an existing database of data for imputation? If not, a standard whole genome method might be more appropriate if you need a more detailed genotype. Is the main goal to identify specific SNPs? If yes, a targeted approach to ensure that your SNP of interest is physically covered might be a better fit. However, if you're fine with inferring the SNP call or the SNP ID through imputation, then low pass could still be acceptable. And then think about how many samples are you going to test. This is where budget and time may play a major part. Screening through hundreds or thousands of samples can be much more affordable and faster using a low pass whole genome sequencing method over a deep sequencing workflow. So now I'd like to highlight a few publications that show how low pass whole genome sequencing can be used. So the first case is from the Broad Institute and was designed to test how effective low pass sequencing and microarrays were for underrepresented populations. Since most arrays are modeled off results from that European ancestry, as I mentioned earlier, there are many variants that can be missed. So the group took data from a deeper sequencing experiment on 91 individuals of African ancestry and downsampled them to data to be comparable to a low pass coverage, such as like 0.5x, 2x, and 4x. This data was compared to the results of different types of arrays, such as low, medium, and high density arrays. Overall, imputation accuracy is affected by the overall sequencing coverage. So the more coverage you have, of course, the better accurate reads that you have. But it is comparable to the arrays that were mentioned before. Now, the low pass option, though, did allow for capture for additional variants that were not available based on the original array design. So this provides an advantage to that study for this specific population. Next, this study coming from a Cancer Institute sought to determine whether low-pass whole genome sequencing could support chromosome instability analysis and how it might be associated with other known genetic prognosis indicators, in particular for breast cancer. So here they were able to use low-pass sequencing to essentially divide the patient tumor groups into two categories, one of high and one of low chromosome instability. Then they took these two categories and then they did a deeper dive using other genotyping methods to determine if there was a link with other known mutations. And they found that there was a high correlation with TP53 loss and also a correlation with how many mutations there were found in the BRCA1 gene. And then lastly, this work from Genentech did test the effectiveness of using a low-pass whole genome sequencing method as a non-invasive alternative to monitor patients with lung cancer. They did this by evaluating the copy number variants of tumor-derived cell-free DNA, and they linked it to how well a patient was performing in the clinic. So patients are being treated with several different therapies, and then they had their plasma collected. And then low-pass whole genome sequencing was then subsequently followed up by a deeper sequencing depending on the variants and the results that were observed in that initial screen. And sequencing was also done to any matched tumor tissue when it was available. So undoubtedly, while higher sequencing depth provided much better resolution and lower background for detecting certain genes and SNP variants, the study did conclude that around 0.5x was the ideal low coverage that was still able to provide comparable calls and the results while minimizing the amount of sequencing that was actually being performed. Based on the samples that had the highest tumor fraction, whole exome sequencing was then performed to further explore any of the mechanisms that might be involved in the resistance to the treatment. So what's really important here is that, cell-free DNA from plasma is often fragmented and does have very low yield. And this group and other groups have actually successfully used this type of material to still be able to predict patient prognosis. So it's one of the many techniques that we hope will eventually be translated into a more common use, even if it's only done as a predictive screen ahead of something that's more established in a clinical setting. Which kind of brings me to, the next stage or what we offer. So the last two cases show how useful low-pass whole genome sequencing can be with performing an initial screen. At Azenta, we have a workflow called the Low-Pass Whole Genome Sequencing Plus that bundles our low-pass whole genome sequencing approach plus a targeted approach. So specifically, this works really well with any large studies that you're performing a deep sequencing, or you want to perform a deep sequencing, but that might not be possible because it's cost prohibitive due to the high number of samples. And you still need to do a discovery, such as looking for new SNPs or something similar. And then, potentially a targeted approach alone, or where an array option might have that bias and you might be missing quite a bit of data. So this workflow not only helps with validating the imputation data, but it provides that more focused interpretation on a potentially smaller data set after this initial screen. So it frees up time and funds to focus on samples or hits that would have the highest return. So in summary, low-pass whole genome sequencing is one of the many techniques that are available for genotyping and profiling. But when deciding which technique to use, we do need to think about what the project goal is and what type of historical data is available to design off of, if it's necessary. Budget should also be considered, especially considering the sample size that you might want to test. For low-pass whole genome sequencing specifically, you do need to think about whether imputation tools are pre-existing, what type of variants or changes you're hoping to capture, and lastly, how large the study is. Low-pass whole genome sequencing is specific to high-frequency variants and is most advantageous for those large-scale population studies that require novel detection. And for designs that fall outside of that, one of the other methods that were discussed today might be a more appropriate fit. So with that, that concludes my portion of today's talk. So thank you so much for your time, and I hope you enjoy the rest of the session.
Thank you for your presentation, Dr. Louie. I am now pleased to welcome Jeremy Lee, who will review the main results from Gencove's research comparing the performance of low-pass whole genome sequencing to commonly used genotyping arrays in the context of Genome-Wide Association Studies and polygenic risk scores.
Thanks, Brianna, for the introduction. My name is Jeremy Lee, and I'm from Gencove, and today I'll be talking a little bit about the paper that we recently put out in the journal Genome Research entitled Low-Pass Sequencing Increases the Power of GWAS and Decreases Measurement Error of Polygenic Risk Scores Compared to Genotyping Arrays. As a bit of brief introduction, Gencove is a biotech startup working on bringing low-pass whole genome sequencing to the industrial scale. As Dr. Louie explained in her talk just now, low-pass sequencing is a relatively new technology that we've been championing that's sort of seen as a next evolution to the genotyping array. And as a bit of personal background, my personal background is in physics and applied mathematics, but my industry experience is in the human genomics. So the primary goal of the study that we set out to publish was to look at how low-pass sequencing compares to genotyping arrays. As Dr. Louie talked about earlier in her presentation, and as many of you might know, low-pass genotyping arrays have been the industry standard for population scale studies that look at common variation across diverse populations, looking to sort of map the underlying genetic architecture of complex traits, which is to say traits that are polygenic or omnigenic in nature, and do a lot more interesting stuff. Basically, sort of the last decade's worth of complex trait genomics has essentially genotyping arrays to thank. However, low-pass sequencing is an alternative method that we've recently championed. This is sort of something that Gencove in particular has been working a lot on recently. Essentially, this is low coverage whole genome sequencing, low coverage, I mean, stuff under 1x, basically as an alternative when combined with imputation as a sort of more modern assay for the types of applications that one would normally turn to a genotyping array for. And so our goal in doing this particular study was to look at how low-pass sequencing compares to genome-wide association studies in particular, and polygenic risk scores, which are two subjects in the genomics world that have been of particular interest within the last 5 to 10 years. And just sort of as a brief review of what low-pass sequencing actually is, this is essentially a fairly simple concept, whole genome shotgun sequencing to a coverage of 1x or below. Typically, we target a coverage of 0.4x or thereabouts. Empirically, that's been sort of the best trade-off that we've seen in terms of sort of cost versus quality of data. Following, shotgun sequencing, we take this data and impute it to large-scale diverse haplotype reference panels. For example, the 1,000 Genomes Phase 3 project, which comprises 2,500 individuals across around 85 million variants. And the output of this low-pass sequencing plus imputation process are high-quality genome-wide genotype calls. And we've previously shown in another publication that low-pass sequencing at a coverage of 0.4x or above actually attains higher power for association studies compared to a genotyping array designed specifically for pharmacogenomics, even in the regions of the genome which are specifically enriched for the genotyping array for sites of interest. And so in this particular study, we aim to look at a slightly different question, which was how does the low-pass sequencing compare to a genotyping array that was designed to assay diverse populations? In particular, we compared the performance of low-pass sequencing plus imputation to the performance of array genotyping using the Illumina GSA, that's the Illumina global screening array, plus imputation to the same reference panel. And to evaluate results across diverse populations, we chose samples from the European and African superpopulations of the 1,000 Genomes project. And across these multiple assays, we compared results from imputed low-pass sequence data and imputed array data using 3 separate metrics, which I'll describe in a bit. And so the study design from a high level is that we took 60 African and 60 European samples from the 1,000 genomes, all of which are well characterized and which have high quality gold standard genotypes available in the form of the very haplotype reference panel that we use to impute. And we use these, we sequence these 120 samples at 0.5x and also 1x on Illumina machines and 1x on and at a coverage of 1x on BGI machines. We were interested in sort of the difference that the library prep and the sequencing machine would make. And we also assayed these same individuals on the Illumina GSA version 3, which is just the most recent version of it, with sort of the most recent global variation that's known and incorporated into that particular physical assay. And once we assayed all these individuals, whether by sequencing or arrays, we then imputed all of these data to the 1,000 Genomes Phase 3 haplotype reference panel, which, as I mentioned earlier, comprises around 85 million variants across the genome, both including SNPs and indels. And once we did this, we looked at 3 particular metrics of interest compared across these same individuals across different assays. We looked at, first of all, imputation accuracy and as measured by the metric of non-reference genotype concordance. This is just the genotype concordance at sites on the genome where the true or gold standard data was not homozygous reference. Basically, this is a measure of how well imputation actually detects variation from the reference, because as all of you know, most humans are the same across the genome. There's variation is on a minority of sites across the genome. And so in order to measure the performance of imputation, it's more accurate or more insightful to look at sites of imputed variation rather than just imputed homozygous reference calls. And the second metric that we looked at was a metric that's directly correlated with the power of any particular GWAS. In particular, this is a metric called imputation R². Basically, this is how well imputed variants correlate to the true variation at a particular site. And this is computed across allele frequency spectrums and across the genome. And we'll look at how this breaks down along the minor allele frequency spectrum a little later in this presentation. And lastly, we looked at the polygenic, the measurement error of two well-established polygenic risk scores in both coronary artery disease and breast cancer. Basically, these are scores that were previously published in the literature, which were well established and well validated on populations of tens of or hundreds of thousands of people. Basically, the application here is that polygenic risk scores are, now a commonly used tool to stratify populations into relative levels of disease risk. Basically, what we were trying to see here was what's the measurement error when actually estimating these polygenic risk scores when using sort of the industry standard genotyping arrays versus low-pass sequencing. And for all these metrics, sort of the gold standard that we compared them against were genotypes from the held out genotypes from the 1,000 Genomes phase 3 project. Basically, what held out means is that when we imputed a particular individual, we held them out from the haplotype reference panel so that the imputation wasn't overfitting in some sense. And so we'll now look at each of these metrics in a little bit more detail. So the first of which was a non-reference concordance. Basically, what you see here is a plot of non-reference allele frequency on the x-axis versus the mean non-reference concordance for that particular allele frequency bin. And here we have the panes are stratified by population, and the colored lines are stratified by the assay that was actually evaluated here. And basically, the relevant thing to notice in this particular figure is the relative performance of all these lines compared to the this sort of grayish green one on the bottom, which is the performance of a genotyping array, the Illumina GSA. Basically, what we see here is that experiments A, B, and D, which are all the sequencing-based experiments, the 0.5x on Illumina machines, 1x on Illumina machines, and 1x on BGI machines, respectively, for the blue, orange, and red lines, all outperform the imputation from the Illumina GSA by quite a bit, especially in the African case, and pretty robustly across the entire allele frequency spectrum. And indeed, one sees that even at the fairly low allele frequencies of 1 to 5%, that the mean non-reference concordance often exceeds 85 to 90%, especially once you get up to the coverage of 1x or above. And so here's a very similar plot. This is the second metric that we're looking at, which is imputation R squared, which is again the correlation coefficient squared or variance explained by imputed genotypes compared to gold standard genotypes. This is evaluated again on a variant level and also can be stratified by minor allele frequency. Basically, sort of how one should look at this plot is exactly the same as how one should look at the previous plot. The left and right panes are stratification by superpopulation, and the color of the lines are stratification based on the experiment. A, B, and D here are the sequence-based experiments, and E, or the green-gray line at the very bottom of each of these plots are the results from the array experiment. Basically, again, we see here sort of a similar pattern in the sense that low-pass sequencing, imputed data from low-pass sequencing exceeds the performance of imputation from the Illumina GSA across the allele frequency spectrum. This improvement is particularly marked amongst the African cohort. And this basically the takeaway here is that even at sort of the target coverage of 0.4x, which is to say experiment A here, we get a consistently higher performing result in at least Africans by this particular metric, and also Europeans at the higher allele frequency on the higher side of the allele frequency spectrum. And lastly, we looked at the polygenic risk score measurement error for across these different assays for two different polygenic risk scores. This particular figure is a little busy, but the point here is that each of these panes is the distribution of measurement error for a given population, African or European, and for a particular polygenic risk score, breast cancer or CAD. And basically, the relevant bit in all of these panes is the relative performance, which is to say where these little dots are compared to this dotted line in each pane. The dotted line is basically the baseline performance of polygenic risk scores as evaluated in genotyping arrays. All the dots below that line are sequencing based samples, which had a lower measurement error. And these little dashes on the very right side of each pane indicate the average performance for each experiment. And basically, the takeaway here is also very similar to the previous two, which is that for the majority of sequencing-based assays, except for this particular unusual case of breast cancer in Europeans, the measurement error in the sequencing-based assays were considerably lower than those in the those as computed from the imputed genotyping arrays, from the data imputed from genotyping arrays. And so again, the takeaway here is that measurement error decreases with increasing coverage on the sequencing-based assay, and also sequencing-based assays in general just happen to have sort of a categorically lower measurement error when it comes to polygenic risk score measurement. And so those are the main takeaways in terms of sort of quantitative measures as regards the performance of these different assays. However, in the real world, when one's actually planning experiments, another thing that actually comes into consideration is logistical concerns. And so, yeah, logistics are important for real-world studies. It's sort of instructive to take a quick look at just the relative pricing of these assays. And so sequencing can basically be, the cost of sequencing can basically be broken down into two main costs. One is the actual sequencing cost, like how many dollars per gigabase. And also the other part is how much it costs to prep a library. And so given October 2020 costs, the sequencing costs on NovaSeq, if you fill the entire flow cell, correspond to $10 for 0.5x, so that's around 1.5 gigabases, and $20 for 1x, so around 3 or 3.3 gigabases. That's just for the sequencing alone. The custom library prep comes in at $6 flat, regardless of the depth of which one is sequencing. And so the all-in cost for 0.5x sequencing comes out to around $16 and $26 for 1x. And both of these basically are list priced. These are list prices, except for this custom library prep that we've designed in-house to sort of be particularly specialized for the sort of sequencing that we do. But in any case, these costs compare favorably against the list price of the Illumina GSA, which has comes out to around $49 a sample. And so with all this in mind, the main conclusions that we came out of this study with was basically that low-pass sequencing we have proposed as an alternative to genotyping arrays for genome-wide association studies for quite some time. We previously showed that in the context of pharmacogenomics, looking at genes that are pharmacogenetically important, that low-pass sequencing already outperforms genotyping arrays, even ones that are particularly designed to assay those variants of interest. In this particular study, we established further that even in the context of cross-population studies, where one is interested in looking not just in Europeans, where the vast majority of data and assays have previously been done, but also in more underrepresented populations, that low-pass sequencing plus imputation remains a superior choice for genome-wide association studies in terms of power, and also polygenic risk scores in terms of measurement error. And with that, I'll wrap it up. I'd like to thank, in particular, the team at Gencove, and in particular, Chase Major, the Director of Operations, Tomas Barisa, our CTO, and Joe Pickrell, our CEO.
Thank you for sharing Gencove's research, Jeremy. I'd now like to introduce Iris Kalka, who will discuss her current work constructing a cohort consisting of 10,000 healthy individuals, all of whom are deeply phenotyped and genotyped using low-pass whole genome sequencing.
So hi, I'm Iris, and as has been told before, I'm a PhD student in Sergel's lab in the Weizmann Institute of Science. Today I'll be discovering novel and clinical molecular markers for disease using integrated genetic cohorts. So as we all know, current medicine for the most case, is one treatment fits all, which means we have a variety of people with the same medical conditions, they all get more or less the same therapy. These individuals will end up having either a positive effect to the therapy or no effect, and sometimes even adverse effects. We'd like to change that, and obviously we're not alone. So current sort of projection for medicine is to move towards personalized diagnosis and treatments, where we have a variety of people, and we don't only look at the medical conditions, but we also look at other markers that they have. For example, DNA sequencing, biome information, blood tests, and the immune system. From that, we derive different therapies to different individuals, and then hopefully have different groups all having effect from treatment. In order to reach that goal, we join a lot of other existing cohorts, and we asked what research cohorts are already being collected today. In this figure, you can see two axes. The y-axis is the phenotyping depth per participant in a cohort, and the x-axis is the number of people in a cohort. Randomized controlled trials, as you can see, usually have very few people and are not deeply phenotyped. Ideally, we'd have a lot of people and a very deep phenotyping for each individual. However, that's not quite possible. So what we do have is electronic health records. And specifically here in Israel, we have Kalka and Maccabi, which are the largest healthcare service providers that encompass almost the entire country. These have very low phenotypic depth. However, they count for millions of individuals each. There's also GWAS known studies or companies such as 23andMe, MyHeritage, which have a lot of individuals, but again, not a very deep phenotyping, with the exception of deep genetic information. Recently, biobanking has also become quite common. Some of the known biobanks include the UK Biobank, the Kaduri, and the Million Veterans. So all of these have more phenotypic information than previously mentioned, but less individuals. Here's where we come in. Our project is called 10K. From its name, you can derive that it's 10,000 individuals, and we're deeply phenotyping each and every one of them in the hope to bridge the gap between these very large cohorts with a lot of information and the missing phenotyping depth. So what exactly are we planning on doing? Each individual in our cohort is going through heart tests such as EKG and other blood tests and other blood pressure tests. They're connected to a continuous glucose monitor for the term of 2 weeks, where every 5 minutes their PPGR is tested. We also collect their diagnoses, both from their medical professional, from their medical professions, but also from questionnaires that they give themselves. We scan their livers using ultrasound, checking for fatty liver and fibrosis. We detect their body composition using a DEXA, which collects the fat distribution along the body, and we also measure their BMI and body circumferences. We monitor their sleep over multiple nights and a longitudinal data information. This gives us information about apnea, snoring, their oxygen and heart rate, as well as the different sleeping stages they go through. They monitor their diet and record everything for at least 2 weeks, sometimes even more. And we also take blood samples, both serum and PBMCs. We record their voice. We connect them to wearable devices. And we perform genetic tests using low-pass whole genome sequencing. In addition, we add RNA expression tests, protein expression, microbiome tests, both gut and oral, metabolites tests, where we test more than 1,200 different metabolites, and we perform an antibody sequencing assay. In this, we test more than 500 antigens. These include pathogens and viruses, as well as allergens and autoimmune antigens. We recently published a paper about the specific antigen assay, where we created a synthetic DNA library and presented the DNA using a phage presentation. And we've added this to different blood samples of nearly 1,000 healthy individuals. Using immunoprecipitation and washing, we discover what are exactly the antibodies each individual has. We've noticed that people are more similar to themselves than to other individuals. We've looked at all the individuals through a timeline of 5 years. On the blue side, you can see the difference between a person and themselves 5 years later. And you can see that the correlation is pretty high. On the red bar, you can see the correlation between an individual and another random individual within that sample. You can see that they're further apart. So we realize that there is a unique signature for each individual's antibody responses that hopefully tells a story about what they've been exposed to over the years. Using all of these different tests, we can profile a lot of body systems. For example, muscles, skeleton, liver, blood, heart, and so on. We've also used a bunch of methods, which I've already mentioned. And the question is, can we use population-level cohorts and this specific cohort for drug discovery? So as Jeremy previously mentioned, other biobanks, such as the UK Biobank, have already performed genetic profiling. And from them, they've learned polygenic risk scores, which imply a disease risk for each individual given their genetic information. We transfer this information to our own healthy cohort, and using the genetic profiling, we predict the polygenic risk score for each individual, having them have a disease risk for whichever disease they might have developed in the future. Once we identify risk groups, we separate the individuals to two subgroups, high genetic risk and low genetic risk. These two groups are then compared to different phenotypes we're collecting. As you've seen, there's many of them. Once we compare different biomarker levels, we hope to find specific biomarkers that separate the groups and thus understand the difference between the two. So why are we looking specifically at genetics to compare these groups? The answer is we'd like to do causal inference, but at the end of the day, we're looking at a single time point for each individual. So we don't really have the ability to see what came before what. This is where genetics come into play, because we realize that genetics has an effect on other markers, especially on the biological markers, in a very direct way. And the causal relationship is clear within that range. We're currently under validation stages. Our first validation was to predict height. And what we were doing is we are validating a projected PRS with measured outcome. So again, specifically, we're looking at height. What we do is we look at PCs of the genetic information, and from it, we try to predict the height of all of our individuals. Then we add a PRS for height that has been computed on the UK Biobank and has been projected on our own individuals. And again, we try to predict their height. We compare these two models to see their effects, and more specifically to see how much the PRS adds to our predictions. So when we ran only on PCs, we see that our Pearson correlation is 0.74. You can see on the y-axis the true height, and on the x-axis the predicted height. What we actually see here is two groups. The left one is females, the right one is males, as you can probably assume. Then, if we add the PRS that has been projected, we see that the groups are more similar, but moreover, we see that the Pearson correlation has risen to 0.79. When we look at the explained variance of this specific model, we see that it is 0.786. which is pretty close to the explained variance from genetics on height. Our second stage of validation was similar to what has been explained before. We looked at CAD and PRS, cardiovascular diseases, and we compared high-risk and low-risk groups. Specifically, looking at the biomarkers, we've looked at the cholesterol distribution between the high-risk and the low-risk groups, and at the hope that the two will differ. This is currently undergoing, which is why I'm not showing you actual results right now. So the question is, where do we go from here? Once we finish the validations, what are we going to do with them? Hopefully, we'll discover novel biomarkers And in order to do that, we have two methodologies. The first one is to separate genetics from environment, find individuals whose genetic risk for developing a disease is extremely high or extremely low, and in reality, their risk of developing the disease using other biological markers is the opposite of their genetic risk. See how individuals defy their genetics and identify the causal reasons for that. Another method is to identify novel associations between phenotypes that we measure and the genetic risk for developing a disease. With it, we might identify new biomarkers that might explain the developing of the disease or perhaps other aspects of the disease itself. Thank you very much, and feel free to contact me.
Thank you for sharing your research, Iris, and to all of our speakers for their presentations. We received some great questions, so let's jump into those. Our first question is for Dr. Louie. Are there DNA quality considerations when using low-pass whole genome sequencing or other methods?
That's a really great question. So short answer, yes, but long answer is it kind of depends. So with low-pass whole genome sequencing, because it's a whole genome sequencing method, it is going to pick up everything that is there and available, but you do have to think of, highly degraded samples. You're going to be missing quite a bit of the genome in that case. So you do have to take poor quality samples with a grain of salt. That said, now, if you do have poor quality samples, using either low-pass whole genome sequencing or even a deeper whole genome sequencing method would be more advantageous over like a targeted method.
Thank you. This next question is for Jeremy. Can low-pass sequencing be used to identify copy number variation?
Yeah, so that's a great question. So I would say this is another one of those, another advantage of sort of the sequencing-based assays over genotyping arrays. It didn't really go into this during my presentation, but short answer is yes. Basically, large scale, when I say large scale, I mean megabase, deletions and insertions can be easily detected using low-pass whole genome sequencing. Basically, anything from sort of small deletions from like say 1 megabase to a whole chromosome arm or even a whole chromosome can be easily detected. Structural variation can also be detected, although it's a little bit trickier. Depends on exactly which type it is. Like it has to be distinguishable from sort of from the type of data that you're looking at. Certain types of translocations can be detected, whereas other sorts of translocations, which basically results in sort of exactly the same biological outcome, cannot be detected. But broadly speaking, CNVs, at the scale that most people are interested in, as relates to complex trait genetics, can easily be detected using low-pass whole genome sequencing. In addition to this, I would also say like the number of copies of not just the autosomal or allosomal genome, but also the mitochondria, have also been shown in the literature to be very useful for like trait association studies, and it's yet another aspect of the sequencing-based assay which basically isn't present at all, or is like a strict improvement from genotyping arrays.
Great, thanks. Our next question is for Iris. What are the main diseases you are hoping to examine?
So for the first part, we're looking into cardiovascular diseases and diseases of the metabolic syndrome. We also have a subcohort of individuals treated for those diseases, which is why we wanted to look at those specifically. And for the other part, especially because of the antibody assay that we're running, we're creating a list of neurodegenerative diseases. All of our individuals are healthy, but might end up developing these specific diseases, which is why we want to look into them.
Thank you. Another question for Jeremy. How extensible are the imputation quality results to other populations besides the ones you presented?
So the sort of big picture answer to that question is that imputation works regardless of population. What it comes down to is sort of the representation in any particular haplotype reference panel. So basically, we're using the 1,000 genomes phase 3. for imputation. Basically, what this means is that Europeans and Africans and African Americans and East Asians are fairly well represented. Europeans and Africans in particular. What are less well represented are populations like Southeast Asian or like East African, Tibetans or whatever. Basically, sort of the imputation methodology and the software and the underlying algorithms, they all work for a given population. What it comes down to is, do you have those in your particular reference panel? And the answer to that in most human studies right now is no, but that's something that we, along with many other groups in academia and industry, have been looking to solve. Is basically collecting and cataloging larger and more diverse sets of genetic variation across the globe.
Thank you. Here's a question for Dr. Louie. What coverage recommendations are required for low-pass whole genome sequencing?
So that really depends on the project goal again, right? So as I mentioned earlier, obviously the more coverage that you have, the higher the accuracy rate. So if you're looking for novel SNPs for new things, or even the copy number detection at a more accurate level, you want to go for that higher coverage range. So for low pass, around that 4x coverage would be like the ideal situation. But if your budget doesn't allow for that, depending on how many samples that you have, you might want to cut back and just kind of see what you get. If you're just looking for known SNPs, you probably don't need that high level of coverage because everything is, well documented in kind of your panels or your databases. So that, 0.1x may be more than sufficient for what you're looking for.
Great. And we'll just take one more question, which is for Iris. What do you see as your biggest challenge in your study?
I think our biggest challenge is defining a causal relationship, because at the end of the day, we only have a snapshot of the individuals. And one of the methods we're hoping to tackle this is that we're collecting actually longitudinal data on individuals. Some of our individuals have already come into a 2-year visit, and we're hoping to continue tracking them to see what they develop and perhaps changes in their biomarkers, so on and so forth.
Thank you, Iris. Unfortunately, that's all the time we have for today. To those with questions we weren't able to answer, our speakers will reach out to you shortly to address your outstanding questions. Again, thank you to everyone who joined us and engaged with our presenters. If you'd like to watch the webinar again, we will be sending a recording to everyone shortly. We hope you enjoy the rest of your day. Bye now.